Hi, and welcome as we start talking about this section's topic, which is prokaryotes. So today we're going to talk a little bit about what is a prokaryote and how they're classified. We're going to describe how prokaryotes vary in their structure and function. We're going to talk about the roles that prokaryotes have in the living world, and then describe how bacteria causes disease. And lastly, we're going to talk about emerging diseases and what they are. So the key vocabulary words you want to pay attention to for this unit, and the first one is prokaryote, which I will definitely explain, binary fission, pathogen, vaccine, and antibiotics. So you'll see those words as we continue with this lesson. So one of the first vocabulary words that we mention is prokaryotes. And what are they? Well, a prokaryote is anything that is unicellular. What that means is basically an organism that has one cell. Humans are multicellular. We have lots of cells that make us up, but prokaryotes are just unicellular organisms, just one cell. The other thing that's really kind of interesting about prokaryotes is they have no nucleus. So if you remember with organisms like humans and dogs, cats, plants, any of those things, those are what we call eukaryotes. So those are the things that pretty much almost any organism that you've seen. And they're identified by having a nucleus, or they have other membrane-bound organelles. On the other hand, our prokaryotes, which we found over here, include our bacteria and archaebacteria. They have no nucleus. So they do have DNA, and that's something to point out. They do have DNA, but their DNA is not located in a nucleus, but just floating around in the cytoplasm of the organism. And usually in bacteria, they're circular, but they're just kind of this DNA floating around inside the cell of itself. Now, what I have shown here are the three domains of life. So every living thing on Earth can be divided into three things. Either eukarya, and a eukarya is anything that has a nucleus, like I said, or we can divide them into a bacteria or archaea. And these are unicellular organisms that have no nucleus, but they're going to be slightly different. I'm going to talk a little bit about their differences coming up. Now again, when we talk about prokaryotes and we talk about the bacteria group, there's lots of different organisms that can fit into the bacteria. But basically, a lot of them are very similar. They're, like I said, one cell. They have their DNA kind of coiled up in here. They do have ribosomes. So that's one thing I just want to point out, because ribosomes have to make proteins, and these cells are going to still have DNA to make proteins. So they do have their little ribosomes to do that for them. But I don't know if you remember about smooth ER versus rough ER. They don't have any ER at all. So all of their ribosomes are floating around in the cell. They also have this cell wall shown here. Animal cells do not have cell walls, but bacteria do have cell walls, which is really a neat thing, because as we're going to discuss later, we'll talk about antibiotics. Those antibiotics are going to target this cell wall. And then they have this outer membrane outside of it too, and they do also have a cell membrane. So that's just a little bit about the typical bacteria structure. Now, besides having bacteria, like I said, you also have archaea is a type of prokaryote. So I kind of put this little Venn diagram for you to talk a little bit about the difference between a bacteria and an archaea. Both of them are prokaryotes, so they have no nucleus, they're small, they have cell walls, they're one cell. But bacteria has a different thing on their cell wall. They have a peptidoglycan. It's basically a peptide and sugar molecules to help make them. That's missing in the archaea. Bacteria have flagella for movement. That's that little like tail that helps them move around. Archaea's DNA, if you look at it, it looks a lot more similar to a eukarya type of DNA, which again, eukarya are multicellular organisms and everything else. And the other thing too is that archaea loves to live in very harsh environments. Here we have like a pool, a hot springs. Archaea is going to love to live in this high temperature. Bacteria can't live at that. So you might see high salt conditions. Some archaea likes to live in there. So you might see some that like to live without oxygen. Some like to live without any oxygen. They might use something like methane as their source of carbon, a sugar source. I had a friend of mine, he used to work with a type of archaea. His was a thing called a thermatoga. Loved to live at high temperatures. That's why the name therma. It was called thermatoga because when you looked at it under the microscope, it looked like it had a little toga on. But it also had to have like low oxygen conditions and high salt conditions, so very extreme environments. Bacteria also have a second membrane that's not found in the archaea or the eukaryotes. So they are slightly different, but they're both part of the prokaryotic world. Okay, so that tells us a little bit what a prokaryote is. Now we're going to talk about how they vary in their structure and their function. Okay, so again, looking at prokaryotes, they can be very, very different ways that they work. One thing they can look at is you can see some of them as being what they call bacilli. Basically, they're kind of like rod shape over here. So these are really zoomed up pictures of them in the microscope. And you can see that they look like lots of rods. Some of them, they have the shape of what's called cocci. Isn't that a fun word to say? But basically, those are all round circles. This one is a spirilla. It has a little more of a corkscrew shape and stuff. So we do identify prokaryotes by things like size, shape, and movement. But we also look at them at things like, do they use a flagella to move? Do they glide on secretions? Those are some other ways that we sit there and kind of identify them. What's really interesting is that sometimes you might get sick and you go to the doctor's office and they test you out and they say, yep, you got a bacterial infection. Sometimes what they do is they might take a sample of whatever bacteria you have and they're going to grow it on a petri dish. So this petri dish is like this little dish that they put the food on. So it's like round and they have food on the bottom. And they might grow some bacteria on top of it. These little colonies of bacteria. And what they will do is they might look at the individual bacteria and actually try to identify it. And they're going to identify it using the idea of the size, shape of how they are. And also they're going to use what kind of cell membrane it has, because some prokaryotes have different cell membranes than others. But that helps them figure out exactly which type of bacterial infection you have, so they know how to treat it in the long run too, which medicines will work best against it. So those are things that do happen maybe outside of the doctor's office, so they can help figure out and help treat you in the long run. Now, bacteria is going to grow very differently than eukaryotes. So I told you that bacteria don't have nuclei, so no nucleus. And here's the thing, is that you probably learned a little bit about cell division. And if you learned about cell division, you learned that cells divided through a process called mitosis. But mitosis really translates to nuclear division. So really what you're doing in mitosis is you're dividing the nucleus. Since prokaryotes have no nucleus, they can't undergo mitosis. What they're going to do is something else called binary fission. And basically what it is that you have this prokaryotic organism starts to get bigger. And one of the first things it's going to do is it's going to take its DNA and it's going to replicate it or make another copy of it. That's what we see here in the lighter blue. There's also things in the cell called, in some prokaryotes, and we'll talk about these a little later, called plasmids. Those are also going to replicate at this point too. Now, the cell's going to start to elongate a little bit. You're going to notice that the DNA is going to start separating out. You're going to get a little cleavage occurring here. And eventually you're going to get two new cells, very distinct from each other, and that's the process of it reproducing and growing. And, lots of bacteria are going to keep growing and growing and growing. And we have ways of killing bacteria. You might be aware of like some antibiotics. You might want to use some cleaning agent. Maybe if you're at home, you use like Clorox or something to help wipe down the bathroom to clean up the bacteria that might be there. But some bacteria don't do well and don't end up getting killed off very easily. And that's an example of one type of bacteria shown here, Bacillus anthracis. Okay, so let me explain what's going on there. So some bacteria, when the conditions get really nasty, whether there's not enough water around, or they might be the bad temperature, too high, too low, or just not a good living condition, what you'd see is that these prokaryotes form something called an endospore, which is shown here. Basically what happens is the DNA gets encapsulated. Let's see if I can put that in a different color for you guys. You get like this little encapsulation of the DNA, and you see that it forms this big kind of capsule around it, and this endospore can stay dormant for a long time. It could be months, maybe even years, and things like that. So what happens is this becomes really hard to kill. You can put a cleaning agent on it, and it won't die, which is really not good. Something like anthrax, which is a disease, basically, is caused by a type of bacteria called Bacillus anthracis that is causing this endospore, and that's why it's so hard to kill. Now, some of you may have seen this machine in one of our classrooms, and Mr. Atchison has it in the back of his room, and this is an autoclave. And what this does is it is used to kill bacteria, and it does a really good job at not only killing the bacteria that is not going into the endospore stage, but also the endospores really well. And it does it by raising the temperature inside here. So we put this, whatever we want to kill in there, whatever we want to sterilize in there, we put the temperature at a high temperature. It's like 121 degrees Celsius. And we also use high pressure. And between the high pressure and the high temperature, we're able to really knock out these endospores and really kill everything. So that's one of the reasons why we have that machine, is sometimes we are making food for bacteria, and we want to make sure the food is nice and clean, so we can only grow what we want. We're going to pop it into the autoclave and sterilize it that way. And you might see this at other places too, whether a dentist office. I think there's also some nail salons use autoclaves to help sterilize their equipment. You know it's getting really good clean because you're killing off all the bacteria you could possibly imagine on it. Now, I don't know if you remember, I told you about the plasmid way back when we were doing budding. So here we have a cell. It has its DNA inside of it. Little DNA. And then there's this little weird thing in there also called a plasmid. I'd say we'll come back to it a little later. Well, plasmids are made up of DNA. But usually they have like one or two genes on it that gives a special identity or a special power, almost, I like to say, to the bacteria. So, for example, you might have this bacteria right over here who doesn't have the special power. In this case, it has no plasmid, it has DNA. But what happens is this particular bacteria is susceptible to ampicillin, an antibiotic. So if you had this bacteria hanging out in your body, making you sick, and you take ampicillin to kill it, it dies off. This little plasmid might have a gene that allows this bacteria to resist ampicillin. It now becomes an antibiotic-resistant bacteria, and in particular, it's resistant to ampicillin. And what happens is sometimes this bacteria can transfer this plasmid to its friend. So what you might see happening is it might form what's called a pilus. It's this little bridge, and when it makes that bridge, it's going to transfer over that little bacteria to go to the other that little plasmid to go to the other bacteria, and now instead of having one antibiotic-resistant bacteria, you have two. I bring this up because this is one of the big reasons why you do have the antibiotic resistance occurring. Is you're seeing that when you have one bacteria that has this plasmid, it's able to pass it on through this process of conjugation to lots of other bacteria, and now you have a whole population that's going to be antibiotic resistant. Now we're going to talk a little bit how prokaryotes are important in our living world. So, we've talked about bacteria, and a lot of people know that we want to get rid of them. They make us sick, right? So we don't like to have them around all the time. But what's interesting is that they do play a really important role in our environment. So some of the things they do is they're decomposers. helps break things down. Some bacteria are producers, which hopefully you remember, those are the organisms that help to give energy to the ecosystem. And some are also nitrogen fixers, which that's important because they convert nitrogen gas into a form that can be picked up by plants, and then that puts it into our food chains too. So really a lot of the food chains that we have are dependent upon prokaryotes as producers of those food and biomass. So again, nitrogen fixation, what really we see is that some of our plants in their roots have these little nodules on there. And if you were to explore those little nodules in more detail, what you would see is that inside those nodules are prokaryotes that have formed a symbiotic relationship with the plant. In fact, there's a type of bacterium called Rhizobium that grows in those nodules, and those help convert nitrogen in the air into a nitrogen compound that's important for that plant growth. So really important process in again, getting plants to grow and also helping getting nitrogen into the food chain. Now I have to also discuss how important the bacteria and bacteria are into human life. We have more bacteria on humans than we do have cells. So what we have learned is that a lot of the bacteria in our body is part of what we call the microbiome, basically just that vast amount of bacteria that helps us to function properly. In fact, if you looked in your large intestine, you would see that there's a milieu of bacteria, and scientists are starting to realize that different people have different kinds of bacteria in there. And so they're realizing that maybe some people are influenced a little bit by their bacteria, maybe for food digestion, might be different than somebody else. And one of the things they're trying to explore is, if this person's having a problem, maybe we can transfer some bacteria from a person who's healthy and get it to that person, and then we can fix it in that way too. So we're really starting to study this. It's a really kind of neat aspect of exploration in science, but it's a kind of neat way of looking at how to help cure diseases and problems that we might see. So that does lead us to basically, how do bacteria cause disease? Well, there are a few bacterial diseases. Basically, a lot of them are disease-causing agents. When something is a disease-causing agent, it is a pathogen. But lots of bacteria are pathogens. And so what happens is bacteria can produce disease in one of two ways. Either it can disrupt the health and cause disease by destroying a living cell, or sometimes it releases a toxin that upsets how the cell's functioning overall. Like, I am almost certain, some of these are here, I'm trying to look at this. I know that we have something like strep throat over here that causes an infection by killing living cells. Versus some other things, some other diseases, bacteria might produce a toxin that's going to cause the cells not to function properly, or something in the long run from that too. So how do we get rid of this? How do we prevent ourselves and control this bacteria? Probably the best way to help control your bacteria infections is something that we all do really well. It's washing our hands. That's going to be really, really effective. That's a form of a removal of the bacteria, but it also helps break down the cell completely. So what's nice about that is that you're getting rid of the bacteria from your hands, but you're also, with the usage of soap, you have to make sure you use that soap, you're going to break the cell membrane enough so that bacteria can't live too. But we can also do things like, disinfectants. Those are going to kill those bacteria. Food storage. Keeping food at a colder temperature is going to slow down bacteria growth. The bacteria is still going to be there, so you have to worry about that too. If you take the cold food and take it out and let it sit for a little while, that room temperature or even warmer is the perfect environment for bacteria to keep growing. So what you're doing is, by keeping it in the cold, you're keeping that bacteria from growing, and so you're reducing the chance of it releasing a toxin that can get you sick. Food processing. So again, there are actually really some neat ways, if you look at the history of even food, whether you're eating, some foods have been made by adding a lot of salt and preserving it that way. I know in my family we like to eat prosciutto, which is a type of meat, and that is processed in a way to kill off the bacteria by adding a lot of salt. The salt actually dries out the bacteria and it keeps it as a meat that can be eaten after being out for a long period of time. I think of something like beef jerky is another form of food processing. By adding some spices and drying it out, you're not making it a favorable environment for the bacteria to grow, and that's why it's kind of preserved. Sterilization by heat. When you cook your food, you're killing off bacteria. So that's another way that you can get rid of the bacteria, is just by cooking it. That's why foods should be cooked to a certain temperature. If you ever get any cooking classes, you'd see that they want the meat to be at a certain temperature. It's to help make sure that you're killing off all that bacteria and preventing people from getting sick based on it. Now, those are all ways of preventing bacteria for especially food processing and stuff like that. But you can also protect a disease from a bacteria by giving yourself the immunity to that disease. One interesting thing that you probably are aware of and know is that you have antibodies in your body. The goal of these antibodies, and they're very specific, is they're to bind to things that are dangerous or bad to your body. And every time you get sick, your body will make antibodies and store it for a period of time, sometimes for a long time, sometimes for a short period of time, so that if you ever get that same illness again, your body can fight it off pretty easily. Well, one thing that scientists were able to figure out is that if you have the antibodies already in there, then you'll be able to fight an infection really well. And so they've been able to make vaccines with either using pieces of a bacteria or maybe a bacteria that was killed off, and they inject it into people. And what happens is that piece is just small enough, not enough to get you sick, but just small enough to get your body activated and say, hey, I'm going to make antibodies to it. So then what happens is you store these antibodies for a long period of time, and if you ever do see that pathogen again, your body already has the antibodies and is ready to fight it off really, really quickly. So bacteria can also be part of emerging diseases. It's not just bacteria. We do also know that some viruses can be part of that too. But what an emerging disease is, it's basically a previously unknown disease that appears in the population for the first time, or sometimes it's a well-known disease that suddenly has become really, really hard to control. And again, shown here, there are some viruses that are coming up here. So these are some of your viruses. Tuberculosis is a bacterial disease. And again, Ebola would be a virus. So some of these are things that we know, like tuberculosis is something we have controlled really well. We have antibiotics to fight it. But now, again, like we told you about the plasmids and the antibiotic resistance, there's some tuberculosis that's coming up that we don't have the drugs anymore to cure it. And so that's becoming a problem. Emerging diseases is still a threat. It's continually being a threat. Things like Zika showing up, that is also another type of emerging disease that recently came into the population. It's new. We don't really know what it does to the body, and they're trying to control it in those ways. So this is where your public health officials are working together to help track it, control it, and find out more that they can to help protect us. But it's stuff that we need to worry about, especially in dealing with viruses and also in bacteria. So that concludes our little chapter on prokaryotes. Hope you enjoyed it.